Nitrogen assimilation is the formation of organic nitrogen compounds like amino acids from inorganic nitrogen compounds present in the environment. Organisms like plants, fungi and certain bacteria that cannot fix nitrogen gas depend on the ability to assimilate nitrate or ammonia for their needs. Other organisms like animals depend entirely on organic nitrogen from their food. Plants absorb nitrogen from the soil in the form of nitrate and ammonia. In aerobic soils where nitrification can occur, nitrate is usually the predominant form of available nitrogen that is absorbed. Most nitrogen is absorbed by plants in the form of nitrate. However, this need not always be the case, as ammonia can predominate in grasslands and in flooded anaerobic soils like rice paddies. Plant roots themselves can affect the abundance of various forms of nitrogen by changing the pH and secreting organic compounds or oxygen. This influences microbial activities like the interconversion of various nitrogen species, the release of ammonia from organic matter in the soil and the fixation of nitrogen by non-nodule forming bacteria. Ammonium ions are absorbed by the plant via ammonia transporters. Nitrate is taken up by several nitrate transporters that use a proton gradient to power the transport. Nitrogen is transported from the root to the shoot via the xylem in the form of nitrate, dissolved ammonia and amino acids. Usually, but not always, most of the nitrate reduction is carried out in the shoots, while the roots reduce only a small fraction of the absorbed nitrate to ammonia. Ammonia, both absorbed and synthesized, is incorporated into amino acids via the glutamate synthase, that is GS-GO-GAT pathway. While nearly all the ammonia in the root is usually incorporated into amino acids at the root itself, plants may transport significant amounts of ammonium ions in the xylem to be fixed in the shoots. This may help avoid the transport of organic compounds down to the roots just to carry the nitrogen back as amino acids. Thus, nitrate reduction and ammonia assimilation are important aspects of utilization of nitrogen by plants. First, we will discuss how nitrate is assimilated in plants. Nitrate assimilation accounts for 99% of the inorganic nitrogen assimilation into organisms. Nitrate reduction is carried out in two steps. Nitrate is first reduced to nitrite in the cytosol by nitrate reductase using NADH or NADPH. Nitrite is then reduced to ammonia in the chloroplast and by plastids in the roots by a ferredoxin dependent nitrite reductase enzyme. In photosynthesizing tissues, it uses an isoform of ferredoxin, that is FD1, which is reduced by PS1, that is photosystem 1, while in the root it uses a form of ferredoxin FD3 that has a less negative midpoint potential and can be reduced easily by NADPH. In non-photosynthesizing tissues, NADPH is generated by glycolysis and the pentose phosphate pathway. In the chloroplast, glutamine synthetase incorporates this ammonia as the amide group of glutamine using glutamate as a substrate. Glutamate synthase ferredoxin GOGAT and NADH GOGAT transfers the amide group onto 2-oxoglutarate molecule producing 2 glutamates. Further transaminations are carried out to make other amino acids, commonly asparagine, from glutamine. While the enzyme glutamate dehydrogenase, that is GDH, does not play a direct role in the assimilation, it protects the mitochondrial functions during periods of high nitrogen metabolism and takes part in nitrogen remobilization. Different plants use different pathways for nitrogen assimilation to different levels. Tomatoes take in a lot of potassium and accumulate salts in their vacuoles. Castor reduces nitrate in the roots to a large extent and excretes the resulting alkali. Soybean plants move a large amount of malate to the roots where they convert it to alkali while the potassium is recirculated. Every nitrate ion reduced to ammonia produces one hydroxyl ion, that is OH minus. To maintain a pH balance, the plant must either excrete it into the surrounding medium or neutralize it with organic acids. This results in the medium around the plant roots becoming alkaline when they take up nitrate. To maintain ionic balance, every nitrate taken into the root must be accompanied by either the uptake of a cation or the excretion of an anion. Plants like tomatoes take up metal ions like potassium, sodium, calcium and magnesium to exactly match every nitrate taken up and store these as the salts of organic acids like malate and oxalate. Other plants like the soybean balance most of their nitrate intake with the excretion of hydroxyl or bicarbonate ion. Plants that reduce nitrates in the shoots and excrete alkali from their roots need to transport the alkali in an inert form from the shoots to the roots. To achieve this, they synthesize malic acid in the leaves from neutral precursors like carbohydrates. The potassium ions brought to the leaves along with the nitrate in the xylem are then sent along with the malate to the roots via the phloem. In the roots, the malate is consumed. When malate is converted back to malic acid prior to use, an hydroxyl ion is released and excreted. The potassium ions are then recirculated up the xylem with fresh nitrate. Thus, the plants avoid having to absorb and store excess salts and also transport the hydroxyl ion. Plants like castor reduce a lot of nitrate in the root itself and excrete the resulting base. Some of the base produced in the shoots is transported to the roots as salts of organic acids, while a small amount of the carboxylates are just stored in the shoot itself. The term nitrification refers to the conversion of ammonium to nitrate. This is brought about by the nitrifying bacteria, which are specialized to gain their energy by oxidizing ammonium while using carbon dioxide as their source of carbon to synthesize organic compounds. Organisms of this sort are termed chemoautotrophs. They gain their energy by chemical oxidations, that is chemo, and they are self feeders, that is autotrophs, because they do not depend on preformed organic matter. In principle, the oxidation of ammonium by these bacteria is no different from the way in which humans gain energy by oxidizing sugars. Their use of carbon dioxide to produce organic matter is no different in principle from the behavior of plants. The nitrifying bacteria are found in most soils and waters of moderate pH and are not active in highly acidic soils. They almost always are found as mixed species communities termed as consortia, because some of them, for example, Nitrosomonas species, are specialized to convert ammonium to nitrite, while others, for example, Nitrobacter species, convert nitrite to nitrate. In fact, the accumulation of nitrite inhibits Nitrosomonas, so it depends on Nitrobacter to convert this to nitrate, whereas Nitrobacter depends on Nitrosomonas to generate nitrite. The nitrifying bacteria have some important environmental consequences because they are so common that most of the ammonium in oxygenated soil or natural waters is readily converted to nitrate. Most plants and microorganisms can take up either nitrate or ammonium. However, process of nitrification has some undesirable consequences. The ammonium ion has a positive charge and so is readily absorbed onto the negatively charged clay colloids and soil organic matter, preventing it from being washed out of the soil by rainfall. In contrast, the negatively charged nitrate ion is not held on soil particles and so can be washed down the soil profile, the process termed as leaching. In this way, valuable nitrogen can be lost from the soil, reducing the soil fertility. The nitrates can then accumulate in groundwater and ultimately in drinking water. There are strict regulations governing the amount of nitrate that can be present in drinking water, because nitrates can be reduced to highly reactive nitrites by microorganisms in the anaerobic conditions of the gut. Nitrites are absorbed from the gut and bind to hemoglobin, reducing its oxygen carrying capacity. In young babies, this can lead to respiratory distress, the condition known as blue baby syndrome. Nitrite in the gut can also react with amino compounds, forming highly carcinogenic nitrosamines. Denitrification refers to the process in which nitrate is converted to gaseous compounds like nitric oxide, nitrous oxide and nitrogen by microorganisms. The sequence usually involves the production of nitrite as an intermediate step. Several types of bacteria perform this conversion while growing on organic matter in anaerobic conditions. Because of the lack of oxygen for normal aerobic respiration, they use nitrate in place of oxygen as the terminal electron acceptor. This is termed anaerobic respiration and can be illustrated as follows. In aerobic respiration, as in case of humans, organic molecules are oxidized to obtain energy while oxygen is reduced to water. In the absence of oxygen, any reducible substance such as nitrate could serve the same role and can be reduced to nitrite, nitric oxide, nitrous oxide and nitrogen. Thus, the conditions in which we find denitrifying organisms are characterized by: first, a supply of oxidizable organic matter, and second, absence of oxygen, but availability of reducible nitrogen sources. A mixture of gaseous nitrogen products is often produced because of the stepwise use of nitrate, nitrite, nitric oxide and nitrous oxide as electron acceptors in anaerobic respiration. The common denitrifying bacteria include several species of Pseudomonas, alkaligenes and Bacillus. Their activities result in substantial losses of nitrogen into the atmosphere, roughly balancing the amount of nitrogen fixation that occurs each year. Plants and microorganisms can make all 20 amino acids and all other needed nitrogen metabolites via transamination or aminotransferase reactions. In these organisms, glutamate is the source of nitrogen. Mammals can make only 10 out of the 20 amino acids. The others are classed as essential amino acids and must be obtained in the diet. The urea cycle, also known as the ornithine cycle, is a cycle of biochemical reactions occurring in many animals that produces urea from ammonia. Ammonia assimilation is the pathway by which ammonia is processed and incorporated into a cell. In an energy rich, that is glucose containing but nitrogen poor environment, glutamine synthetase and glutamate synthase form an ammonia assimilatory cycle in which ammonia is incorporated into L-glutamate to form L-glutamine, which then combines with alpha ketoglutarate to regenerate L-glutamate. This ATP dependent cycle is essential for nitrogen limited growth and for steady state growth with some sources of nitrogen. Ammonia is incorporated into amino acids via the glutamine synthetase glutamate synthase pathway. While nearly all of the ammonia in the root is usually incorporated into amino acids at the root itself, plants may transport significant amounts of ammonium ions in the xylem to be fixed in the shoots. This may help avoid the transport of organic compounds down to the roots just to carry the nitrogen back as amino acids. For many years, it was thought that bacteria and higher plants assimilate ammonia into glutamate via the glutamate dehydrogenase pathway, as in certain fungi and yeast. However, in bacteria, it became clear in 1970 that an alternative pathway of ammonia assimilation involving the enzymes glutamine synthetase and an NADPH dependent glutamine 2-oxoglutarate amidotransferase or glutamate synthase, which must be operating when ammonia is present in the growth medium at low levels. Thus, nitrogen starvation leads to de-repression and activation of GS, which has a high affinity for ammonia, and de-repression of GO-GAT and repression of GDH with a relatively low affinity for ammonia. High ammonia availability leads to repression and deactivation of GS and induction of GDH. Both the GDH and GS-GO-GAT pathways produce 1 mole of glutamate from 1 mole each of ammonia, 2-oxoglutarate and NADPH. But note that the GS-GO-GAT pathway is energetically more costly than the GDH pathway, consuming 1 ATP. Please note that glutamine is the nitrogen donor in synthesis of many nitrogen containing compounds. Nitrogen enters roots as nitrate or ammonium ions. The ammonium ions is incorporated into amino acids in roots and leaves, and the amino acids accumulate in proteins. The main, if not sole function of some proteins is to provide a store of amino acids. Ammonium ions formed as a result of nitrate reduction or nitrogen fixation is first incorporated into amino acids via the glutamine synthase reaction. The glutamate consumed by the GS reaction is replenished by an alternative mode of glutamate synthesis, that is GOGAT. Plants contain multiple isoenzymes of GS, some located in the cytoplasm, that is GS1, and others in plastids, that is GS2. All plants examined contain at least 3 GS1 isozymes, but only one nuclear encoded GS2 isozyme. GS2 appears to be the most important in ammonia assimilation, but GS1 isozymes are also functional. In C3 plants, ammonia production from photorespiration can exceed that formed by nitrite reduction by 10-fold. Nitrogen, following its initial assimilation into glutamine and glutamate, can be distributed to many other compounds by the action of aminotransferases. The final step in the synthesis of several amino acids is the transamination of the 2-keto analog of the amino acids. Glutamate is often the amino donor substrate in biosynthetic reactions, with the result that 2-oxoglutarate will be a product. 2-oxoglutarate is a substrate required for nitrogen assimilation by both GDH and GS-GOGAT pathways. Glutamate utilizing transaminases therefore regenerate the carbon precursor needed for ammonia assimilation. Regeneration of 2-oxoglutarate may permit assimilation to continue in the absence of net 2-oxoglutarate synthesis as long as glutamate is not utilized by other reactions. This enzyme is very strictly regulated. In bacteria, it is regulated in 3 ways. First, by repression-derepression mediated by glutamine. Second, by cumulative feedback inhibition, which is inhibited by end products of glutamine metabolism. And third, by nucleotides such as ADP and AMP. They inhibit by reversible deactivation, that is by adenylation of GS in gram-negative bacteria. In fungi also, this enzyme is regulated in 3 ways. First, by repression and derepression mediated by glutamine. Second, by cumulative feedback inhibition, where GS is inhibited by end products of glutamine metabolism. Third, by reversible deactivation, where there is dissociation of active octamers into deactive tetramers. In higher plants, regulation of GS takes place in 2 ways. First, by cumulative feedback inhibition, where it is inhibited by end products of glutamine metabolism, and secondly, by nucleotides such as ADP and AMP. There is limited evidence for repression and derepression mediated by glutamine in higher plants. Thus, in this module, we have seen how atmospheric nitrogen, which is first converted into ammonia, is then utilized to make important compounds like amino acids. Glutamine is the major amino acids in which nitrogen is stored and then transferred to other amino acids by transamination reactions.